Okay. So splicing, obviously, a very important type of thing, and binds to generate numerous different RNAs from the, or rather, numerous different messenger RNAs from the same type. Now here again, in many cases you'll have a protein well, let's put this down. Okay, most alternately spliced RNAs are going to generate a family of related molecules. In other words, you have a cell-specific version of this protein in this cell type, another version in a different cell type, and so on. But they'll have basically the same general function, just specific differences. That's the majority of things we see here. But you can sometimes get some significant changes. And we're going to see, this is going to be after the exam, but we're going to see some examples where you take one RNA, splice it one way. Let's suppose it codes either for a transcription factor or a splicing factor. You splice that RNA one way, and you have something that activates transcription or splicing. Splice it a different way, we get a protein that binds the same site, but it acts in the opposite, is an inhibitor or repressor. So we can say, so alternate splicing can generate transcription or splicing inhibitors or activators. Splice it one way, it is a repressor or inhibitor of that. Splice another way, it's an activator. Now that can be useful. Let's suppose you're one cell type and you absolutely positively need to, say, transcribe this gene, transcribe gene A, but do not, under any circumstances, transcribe gene B. And then a different cell type wants to transcribe B but not A. You could use the same transcription factor. Splice it one way, it's a promoter of transcription of gene A and a repressor of gene B. Splice it a different way, it is a repressor of gene A and a promoter of gene B. Same protein, same gene, different proteins generated by alternate splicing. We'll see examples of that later on. Yes, it looks like you got a question.
Yeah, how much of this splicing is going to be on the test?
Okay. Not much. Most of this is going to be on next exam. Almost everything that we covered on Thursday, that will be on the exam. This is a little bit above. There's one or two very general questions on splicing on this exam, but not this level of detail. So this is going to be second exam. This is second exam. Okay. All right. So that's another kind of thing. Now, occasionally, okay, alternate splicing can sometimes generate, it's not common, but sometimes you can generate radically different molecules. For instance, there's one gene, I forgot which one it is, but the precursor of the messenger RNA gets spliced in one way in one cell type, and it becomes a neurotransmitter, a small protein neurotransmitter. A different cell type, it gets spliced in a different way, and it actually acts as a hormone stimulating various developmental processes in certain cell types. So you have something that has very different functions, even though it all came from the same gene. that usually requires pretty dramatic alternate splicing. Like, you know, we're going to have 10 exons, and we're going to keep, this cell type keeps 1 through 6, and this cell type keeps 4 through 9, you know, and so on. So, but you can sometimes get different proteins. It's not as common as just having a family of cell-specific related proteins, but it has been known to happen. We do see that. Okay, that's a couple things about alternate splicing. We have a great deal of complexity here. Now, once again, when you look at up the sort of evolutionary tree and stuff, yes?
Was those the roles?
Yeah, those are examples of what we can do with alternate splicing. Most common is generating a family of related proteins that have basically similar functions, which is specific variants. Less common, but you still find something that's an activator when spliced one way and a repressor when spliced the other way. Still less common, but it's been known to happen, is proteins that are wearing completely two different hats, totally different cellular functions, even though they're coded for by the same gene. Okay, now, what we see is you don't get much in the way of alternate splicing, what we could consider, quote, lower animals, things like roundworms and stuff like that. But we start getting into higher organisms, alternate splicing becomes more and more common. That's why our genome, that's why fruit flies have fewer genes, have only about 2/3 the number of genes that little tiny roundworm. In fact, there are some protists like slime mold and some of the complex ciliated protists. Those guys have as many genes as the fruit fly does. That's because there's a lot of alternate splicing in fruit flies, even more in us. It's estimated that probably estimated that at least 60%, and it could be a lot more. This is still like, they keep on upping the number every time I see a review on this stuff. So let's say 60% at the minimum of our genes are alternately spliced in one way or another. Okay. I'm going to guess that the ones that don't get much alternate splicing are for real basic housekeeping genes. You don't want to have a different version of a protein going into the ribosome too often. You don't want to have a different version of the proteins that are found in the electron transport chain in the mitochondria, because it might not work very well, stuff like that. So that's probably a minimal figure. Now, our estimate of, say, if our estimates that we have a little over 20,000 genes, but we have 400,000 or 500,000, perhaps as many as 1 million distinguishable proteins in our body, that's telling us something. That's telling us, on the average, each gene, by alternate splicing, codes for 20, maybe even 40 or 50 different versions. That's quite a bit. Now, obviously, it's not true of every single gene. There are some genes that we see only a couple different variants. But there are other genes that there are hundreds, if not in some few cases, even thousands of different variants, all from the one gene. So that's something. That makes us have a nice, at least in terms of protein coding genes, we have a nice, fairly compact genome. There's an evolutionary reason for that, by the way, because if you have a lot of genes, a huge amount of DNA that's all different kinds of genes and stuff, you have a bigger target. It's easier to hit a big target with chemicals, radiation, what have you, than a smaller target. So if you've got hundreds of thousands of genes, it's likely that you're going to have a high mutation rate compared to not having. As a matter of fact, before they actually sequenced the human genome, there were estimates. They even had a bunch of people that were doing this at a betting pool as to how many genes a human being has. I think they all lost. It was a lot less than they expected. But your typical estimates based on how what the mutation rate was between 50 and 100,000. It turned out that was actually less than 25,000. That stunned people when they started counting genes, but that seems to be the case. But at any rate, that way you can have code for a lot of proteins and a relatively limited number of genes. Now, one last little point here is, although the majority of genes get alternate splicing, there are one set of genes that never get alternate splicing. They don't even have any introns. It's a single coding sequence. Look, no introns. And those genes are the histone genes. The histone genes are a single coding sequence with no introns because the sequence is so similar from one organism to the next. It's so finely tuned, even comparing protist histones and human histones, at most you have 1 or 2 amino acid substitutions. Usually one type of amino acid, like a polar one, small polar one, for another small polar one. Okay, so what that means is histone genes, don't mess with them. Change them in the least and they won't work properly. We saw all the roles of histones, not just for packaging DNA, it's regulatory roles, epigenetic roles, all kinds of things. You start messing with the histones, you're playing with fire and you're playing with death. Okay, so there's no need to do alternate splicing because any version of histones is probably going to be non-functional. And you don't want to take the chances that you might accidentally get splicing mistakes and make histones that will misfunction. They may bind DNA, but imagine a mutant histone that joins to form a nucleosome and binds DNA, and it's missing a critical acetylation or phosphorylation site. And lo and behold, you want to transcribe that gene, and the kinase or acetyltransferase comes up there, and I'm going to stick a phosphate on it. You say, don't hold on to the DNA as tightly as you do. And it's like, where's the site? It's gone. It's gone. Can't do anything about it. So now that nucleosome stays tightly bound to DNA, and lo and behold, guess what? That gene doesn't get transcribed. Not good. So, histone genes, they're going to be hyper-conservative here. You cannot afford mistakes, so we're going to play things as safe and conservative as possible here. Okay, well, when we come back on Tuesday, after suffering on Thursday, when we come back on Tuesday, we'll see a few specific examples of alternate splicing. We illustrate the principles of strong release splice sites, splicing promoters or activators, and splicing repressors. And we'll see that in action using a couple specific examples. So, I'll see you guys in the lab. I've got a few things, once again, the usual kind of thing. I can set the lab quiz up. So, I'll see you guys fairly shortly.